Thank you very much, Joe. We're going to begin with a panel discussion now, and I would like to remind those watching on the webcast that you can submit questions through the Q&A window. We will get to those questions toward the end of this panel discussion, and I know that we've been collecting some already. But I wanted to start with building on what Joe was just describing and picking up on a theme that brought up by all of our speakers this morning related to the role of unique collaborations in solving some of the problems that have made it difficult to succeed and have resulted in high failure rates in late stage development for chronic prevalent diseases. Joe has just described a public-private partnership model and the programs that they're working on in some of these prevalent chronic diseases, but I presume the capacity of the FNIH is only so great, and there remain many, many more chronic diseases that require greater understanding of biological targets, more biomarkers that can possibly be used as surrogate endpoints to reduce the risk or improve decision-making in clinical development. What's the role of collaborations and partnerships, and are there models in addition to the type of work that FNIH is doing that can be brought to bear on solving some of these problems in prevalent chronic diseases? I'll ask you to start, Russ, because I know this is something that you've talked to the patient and caregiver community and about Alzheimer's disease. How else are you thinking about involving other stakeholders in collaborative problem solving in these areas?
So thanks for the question. The research I referred to, where we asked patients what matters most to you, symptoms that you'd like to avoid, capacities that you'd like to keep, was actually done in a pre-competitive consortium of largely pharma. We had some private money in there as well. Put some of our own money from Us Against Alzheimer's in there to do some pretty rigorous research. So getting at just plain, we spend a lot of time in this world thinking about druggable targets and molecules and cells. In the end, what you want to do is you want to make people's lives better. And so getting to the very definition of what am I going to see that makes my life better? We did that in a pre-competitive consortium. We are thinking that there's probably room for a real-world evidence initiative that is pre-competitive, that would not, wouldn't prove a whole lot, but would generate a whole lot of hypotheses and would potentially help us. I think I'm forgetting now which of the panelists talked about sort of identifying what drug might work best for you. I mean, we know in cancer, since I was a kid, cancer was a thing. Now we know cancer isn't a thing; it's a whole bunch of things. To what Jason was saying, I suspect with Alzheimer's, we're going to discover that there is no one thing; that there's a collection of things. And as we get more and more personalized with this medicine, we think this pre-competitive group coming together on real-world evidence. Lots of people have lots of data. It's valuable to them, but they all have slices. We think maybe linking the slices is the next big thing for pre-competitive collaboration around Alzheimer's.
Thank you. I want to ask the question of Jason, but also with a twist. So as Russ has just discussed, getting input from patients and families about what's really important and what is really life-changing is an extremely powerful tool for establishing research priorities and identifying where we need more tools and more knowledge. And I want to link that to something that you brought up in your presentation, Jason, which is the question of diversity and how the lack of it is affecting not just the enrollment in clinical trials, but when we think about the lack of diversity, even in our biorepositories and other crucial tools that inform the early research, how can we work through collaborations and partnerships to address some of those problems so that the inputs earlier on are more reliable and more usable for more patients?
It's an excellent question, Susan. And I think there's a multifold answer to it. So one of the startups that I highlighted in my presentation, Egality, that's working towards clinical trial diversity, I spoke to the founder recently, and one of the things that she highlighted to me is that it started with her engaging with community groups because she wanted to go and understand what their concerns were, what their perception was, because it's not just a push from the market saying we need to increase diversity. The FDA now has a policy out saying diversity matters in clinical trials, for example. It was also misunderstandings and deep-seated mistrust from years, decades, centuries even, of abuse of some of these communities. So I think the same thing goes when it comes to biobanking. I mean, there's still the scars of Gila and Henrietta Lacks and that scandal for, how certain underserved populations have taken advantage of us for scientific purpose in the past. So as much as we want to go forward and say, wouldn't it be wonderful if we had increased representation of biobanks, if we started to include more diverse groups in our trials and our preclinical development pipelines, a lot of those groups feel scarred, they feel hard done by. So you have to engage with them on that personal level and understand that, as well as encouraging them to say, now take this COVID vaccine, trust me, it'll save you. Or yes, please do sign up for this trial. I've got a drug that'll address a need that's disproportionately affecting, let's say, Black and brown people, or disproportionately affecting women. It cannot just be you wake up one day and decide that it's time to put equality at the forefront because it's either good for business or it's good for PR. You have to address those long-seated issues. And I think there needs to be a discussion around how those communities are incentivized to participate and actually benefit from the discoveries that are being made off of the back of including them in some of these pipelines. I cited Henrietta Lacks just now. There was a case in the UK where there was a quick example. A young woman presented in the hospital. She had hematoma. They found out that she actually had an antibody in her blood that prevented her blood from clotting, although she was presenting with clots. And it turned out that that drug was turned into an antibody to exocyte 1 and thrombin, and was sold for, I think, several hundred million to J&J. Now, you should ask yourself, how much went back to the young woman who presented with that antibody? I will leave you to come up with the answer to yourself about how much you think she was compensated. So I do think there's also another way we should be looking at the model for how we engage with patient advocacy groups, how we engage with these communities, and how they can feel not only can they trust the science, that they'll benefit from the science, but hopefully that financially we'll be investing in programs of education to increase diversity in the boardroom, on the science teams, on the clinical trial teams, in the R&D teams that are actually developing the next generation of drugs and compounds. I think only then can we address the kind of systemic discrimination that we have been seeing plaguing the development field of pharma and therapeutics from day one.
Susan, I wonder if I can build on Jason's thing for just 1 second. It's not only diversity within, say, the U.S. or the U.K. It's globally, right? If you look at you talked, Jason, a minute ago about GWAS studies. If you look at genome-wide association studies that relate to the field I know about, Alzheimer's, almost all of that is done in what we used to call first world countries, and very little is done in the developing world. Genomes don't know where they live. There's a lot we could learn if we would diversify that kind of work as well.
Thank you. I think there's certainly no question that by not diversifying, we're missing out on a lot of knowledge that could impact success rates all throughout the stream of research and development. One of the things I want to sort of shift topic a little bit, although it is related, is there's a tension in R&D for prevalent chronic diseases between a need to better understand the disease itself, and to invest in more fully understanding the underlying biology. And then the drive toward developing more personalized treatments where you're digging deeper and deeper into subgroups that or even eventually, to individuals. And Joe, I want to start with you on this topic. What's a way to think about balancing those needs, and how to focus the efforts and the investment on those sort of ends of the spectrum?
Yeah, it's a great observation, and I've witnessed it actually personally myself at meetings when I was at pharma, where we would be talking about a particular drug approach, and we would say, well, we have this focus on this particular subset of patients with disease X. And we know that we can, really focus on them, and having the senior, senior leadership say, that's, great. Do we really have a good biomarker? Do we have a really good way to separate out those people? And then in the course of the hour-long meeting, 20 minutes later, have those same leaders say, but we really want a drug that'll hit all of them, right? All of disease X, right? And so, talk about having kind of a difficult time with that. So even from a decision-making point within at least big pharma, the mind has not been made up. We have not made up our mind how we're going to do that. We see successes in cancer, which we continue to hope will drive us to a point where, other diseases are recognized as, if there is a subtype of type 2 diabetes that I can really nail with a particular therapeutic, that will be a profitable enterprise. Until we can get to that kind of comfort, it's going to be an uphill battle for all of the people in the research space within big pharma. I think there are companies, as there are companies that are out there that are focusing very much on rare diseases, and they're making a living, right? So I think that it's just in some ways a matter of time, or one or two really big successes from those little companies in chronic prevalent diseases for the rest of the field to pick up and go. I know the folks that we work with, they are very much interested in very specific and personalized medicine, but they still have to have a drug that goes along with it. And so, it's a difficult question. I'll tell you the other thing that makes it difficult. This is Joe Menetski's point of view. I think we've spent a lot of time in the past characterizing diseases as big things. And Russ pointed out Alzheimer's disease, which is probably more than one. Type 2 diabetes is probably type 50 diabetes. All of these things, because it's convenient, right? I mean, that's what we put them into a group like that, because it's convenient for someone to understand, oh, I have Alzheimer's disease. But in reality, they have one of the 10. And it would be, it also adds complexity, because now if you do come up with a drug that helps 10% of Alzheimer's disease patients, and you know it. You know that you have this drug that will nail 10% of them. The 90% are going to like, well, what about me, right? And so it's not just, it's an endemic concern problem across the board. And one of the reasons we spend a lot of time with patient groups and others is because it's really important for patients to understand that if we go to personalized medicine, it might not be you, right? And that's been kind of learned, as you mentioned, in cancer, right? Cancer, they understand it's not, even if you have prostate cancer, you don't have, it's not all the same thing.
Right.
Anyway.
Yeah, I think we are learning that from cancer, and it's likely that there are lessons from how this is playing out in cancer that we will be able to apply to other more prevalent chronic diseases if we can just crack that first success, as you said. I want to make sure that we have some time for the audience questions, which I know that Amanda has been collecting for us from the Q&A box. What does our audience want to know?
Yes. So the first question that came in is they're curious for some thoughts on whether it's possible to split products between diagnostics and developing new medications if the disease in question has a need for both. So I think the question is maybe about how you would approach developing two, like basically biomarkers for two different things, for diagnostics and for maybe monitoring disease progression or other things that would be relevant for medication development.
Jason,
So,
I'm sorry. Go ahead, Joe.
No, I'm just going to say there biomarkers tell you what's happening in the disease. They don't have to be a target, and in fact, frequently are not. And so they always are separate things. And in terms of a biomarker, there are multiple biomarkers for any one drug development program. You have biomarkers to understand whether you're hitting your target. You have biomarkers to understand whether whether the patient population is correct, whether the disease is getting worse. Each of those is a totally separate thing. And so for every, and for those who are involved in clinical trials, you have safety markers. They have nothing to do with it, but that's, you need them. Right? You can't do without safety markers. So I think there's already an understanding that you need multiple markers. And then on the other side, you need targets that are influential on the disease. And that's a different thing.
So, and I think also if we sort of look across the historical pipeline and again, taking many examples from development of diagnostics and in particular companion diagnostics in the cancer field, I guess it really depends on what you're looking for and what you're trying to show, right? So we see companion diagnostics that have been developed closely alongside a drug candidate aimed primarily at identifying a population of responders to that drug candidate, which is a different thing from finding a good and sensitive and reliable diagnostic for Alzheimer's disease, and is a different thing from the example that Jason referred to in his presentation with Cologuard from Exact Sciences, which is developed as a standalone product for a broad screening application. But I think that maybe Jason, since you referred to the Cologuard example, can you talk a little bit about your perspective as a funder on the importance of diagnostics as products and how that relates to improving success in early development and research for chronic prevalent diseases.
Happy to. So before I became an investor, I was actually in a diagnostics company, one that started off selling research tools and pivoted to diagnostics off of the excellent advice of Grace Colon, who's one of the speakers at the seminar, so you'll be hearing her shortly. But I am very much a proponent of the importance of diagnostics, particularly at the earliest stages of development. When we were at Cambridge Epigenetics, we had the potential of not only looking at genetic mutations or germline mutations, somatic or germline for cancer, but also looking for a wide variety of diseases where epigenetics mattered. So you started to see, is there a possibility of detecting some diseases earlier? Could you stratify better for therapy? Could you monitor minimum residual disease or monitor response? So I think that they should go hand in hand, particularly when it comes to the next generation of therapies that we're seeing today. There's no excuse with the number of omics and platforms available that would allow anyone developing a new drug modality to say that they are going to look at diagnostics as an afterthought. I think also we're increasingly going to be seeing early detection and screening, but there is a caveat. So the Cologuard example is a fantastic one because the standard of care, if you get a positive test, what happens next? You go for a colonoscopy. And chances are, if you catch the, let's say advanced adenoma stage or stage 1, even stage 2, you can zap the cancer and you have really positive outcomes. If you have an early detection or diagnostic test and there's no follow-up, it's just you may or may not have this disease and there's nothing that comes afterwards, the value is a lot harder to convince for the reimbursement landscape, side of things, as well as the investor side. You need to have some standard of care pathway that you're feeding into where you can say, this diagnostic matters. So there's always that caveat, I think, with diagnostics. What is the consequence if you have a positive or negative result?
That's an excellent point. And so we do often see that's sort of interrelationship between the availability of treatments and an investment in diagnostic development. Amanda, do we have another question from the audience?
Yes, I'll ask one more, although we have, I think, many more comments and questions than we'll have time for in this panel. So one came in from Klaus Romero, who asks if Jason can comment more on the value of having a proper handle on uncertainty as a component of decision-making for potential investors, such as decision science and understanding sources of variability.
I will keep it very succinct. I will say that it's extremely important. And the reason being is that we're all human at the end of the day. We suffer from things like confirmation bias. If you see somebody who's pitching an idea to you, you say, oh, I really like them because they're just like me. Or that's an area that I'm familiar with. It must be good. Or if only they did X, Y, and Z, my investment will fly and will save so many lives. We have to have objective measures of, let's say, the likelihood of success and the impact that a particular investment and platform or solution will have. So it's very important that we do not simply rely on our own personal biases, but that we seek external advice and have hard, cold metrics for analyzing the investment opportunity.
Excellent. And with that, I think that we are out of time for this session. I would like to thank very much our speakers, Russ, Jason, and Joe, for sharing their perspectives on these topics. And I look forward to the remainder of the discussion in the workshop today, where I know that we will be getting more in depth into questions related to the use of biomarkers and registries and new technologies enabling discovery research in prevalent chronic diseases. Now we are going to adjourn the workshop for a 30-minute break. Amanda, are we still returning at 1:05, or we, as we're 5 minutes behind schedule, are we going to delay that?
We actually had a little bit of extra time built into the agenda already, so we're still on time for everyone to return at 1:05. Eastern
Great.
time,
Well,
1:05 Eastern.
see everyone at 1:05 Eastern. Thank you all very much.
Thank you.